Hi, students. Welcome back. Today we're going to take a look at protein synthesis. At this point, we know about the DNA molecule as the molecule that's responsible for the inheritance of traits. But it's one thing to know what the molecule is, it's another thing to know how do you get the traits that you have from that molecule. And that's what this section is about. The relevant reading can be found in our bio book, Chapter 10, sections 10:6 to 10:15. Let's start with this very central question. How are genes, that is, the traits that get passed from parents to offspring, and proteins, the major structural components of our bodies, related? Knowing that DNA is the molecule of inheritance isn't all that useful until it gets expressed. Proteins are responsible for the vast majority of a cell's structure and function. It's the material that makes up the cells, the membranes and your skin and fingernails and hair and bones and everything in your body is made up mainly of proteins. Okay, so if DNA contains the information that a cell has, somehow it's got to be linked to the proteins. Well, there's a whole other type of nucleic acid. It's called RNA. RNA stands for ribonucleic acid. Whereas, if you remember from our notes last week, DNA stands for deoxyribonucleic acid. The ribonucleic acid, or RNA, serves as the intermediary, that is, the molecule that goes between DNA and proteins. The RNA carries information from DNA that resides in the nucleus for eukaryotic cells and carries it out to the cell organelle that is responsible for assembling proteins, the ribosomes. So it's as if the RNA takes the information from DNA and acts as the messenger to run the factory that is the ribosome. If you're running a factory, you've got to have instructions for how to make whatever it is you're designed to make in that factory. Well, the instructions are the RNA molecule. Okay, so RNA and DNA. Let's draw some comparisons. They're both nucleic acids. Na stands for nucleic acid. The only difference would be this. Remember, DNA is a double helix. It's a double-stranded molecule that twists upon itself. Well, RNA, it's only one strand. There's no complementary base pairs in RNA. There's only one strand. RNA, by its name, ribonucleic, as opposed to deoxyribonucleic, contains the sugar ribose instead of deoxyribose in the case of DNA. And then thirdly, the bases of DNA are adenine, thymine, cytosine, and guanine. In RNA, there's no thymine. Instead, there's a base that's closely related structurally called uracil. Uracil takes the place of thymine as a nitrogen-containing base in RNA. Take a look at these two molecules. On the left is ribose sugar, and on the right is deoxyribose sugar. This is a bit like one of those games that you see. Spot the differences. Can you see a difference between these two molecules? It's kind of hard to see. I'll give you a couple seconds to look. In ribose sugar, there's an oxygen here at this bonding position. In deoxyribose sugar, there's no oxygen there. That's the only difference. Other than that, they're exactly structurally identical. So RNA and DNA, gee whiz, they're very, very closely related molecules. Okay, we said that RNA was the intermediary between DNA and proteins. Take a look at this picture. This is sometimes called the central dogma of molecular biology, meaning it's a really, really important part of understanding how the biology of our cells work. Start on the left side with DNA. DNA does make copies of itself in the process called replication, and we will study that in a future unit, next unit actually. DNA can also be made into RNA, or rather its message is transcribed in a process called transcription into an RNA messenger. The RNA messenger can then be made into proteins using a process called translation. Transcription happens in the nucleus. Translation of RNA into proteins happens at the ribosome. In order to get from the nucleus to the ribosome, we need the instructions, the RNA molecule. We call it the messenger RNA molecule. We'll take a closer look at that now. Okay, the major steps. First, transcription. Your DNA molecules in the nucleus contain lots and lots of information, thousands upon thousands of genes, which are made up of hundreds of millions of base pairs in the really, really long DNA molecule. And by the way, you have 23 types of chromosomes in every one of your cells, and 2 versions of each of the 23 types of DNA. We'll get to the genetics part later. So there's a lot of genes. Transcribing genes means copying information from DNA into a mRNA molecule. mRNA stands for messenger RNA, because there are other types of RNA, as we'll see. So M for messenger. Secondly, translation. Using the information from the messenger RNA molecule to make a specific sequence of amino acids. Think back to our unit on organic molecules. The subunit of all proteins are amino acids that are going to be linked together by a peptide bond. When you bring a certain sequence of amino acids to a ribosome, they get joined together, kind of like making links in a chain, a long chain sometimes, many thousands of amino acids for a typical normal sized protein, but you've got a lot of them. Take a look at this picture. Here's a double helix molecule, a double-stranded, twisted around together molecule. But in the middle, it's unwound. There's a special enzyme that helps to unwind the DNA so that it can be transcribed. We don't need to know the names of all of the important enzymes, but this one that's listed here is important. The one that transcribes the DNA to make messenger RNA is called RNA polymerase, meaning it's going to make a polymer, a many unit molecule of RNA. And since it's an enzyme names almost always end with ASE, ACE. As the RNA polymerase reads the bases from one of the two strands of DNA called the template strand, the one that you're going to copy, it's manufacturing an RNA single-stranded molecule from it. So the action of the polymerase in this diagram is from left to right, as it does the business of copying the template strand into a single-stranded RNA molecule. You get a single-stranded molecule coming off. At some point, the RNA polymerase is going to read a portion of the DNA that says, stop transcription. Don't copy anymore because you've reached the end of the protein. It may be a long, long, long chain of nucleotides before it reaches the stop signal. But at some point, if you're not going to transcribe the entire DNA molecule, you've got to have a stop signal. That's an important part of transcription. The amino acids that make up proteins are coded for by 3 consecutive DNA bases. Remember, in DNA, there's adenine, thymine, cytosine, and guanine, 4 different nucleotide bases. If you have 3 positions in your code for signaling which amino acid gets brought to the ribosome, we can code for, well, let's see. Well, there are 20 different amino acids. This is a little statistics question, so if it's not your bag, that's quite all right. 4 nucleotides in the first position times 4 nucleotides that could occupy the second position times 4 nucleotides that could exist in the third position. Little bit of math here. 4 times 4 times 4, which is 16 times 4, gives you 64 possible codes. Now, if you had just given 2 consecutive DNA bases, you would have 4 times 4, or 16 options in your code. If it's a 2-letter code, you only can make 16 combinations. Well, students, there's 20 different amino acids that exist, so we needed a 3rd position in the sequence of DNA bases to make a code. All right. When transcribed into messenger RNA, the 3-letter codes, or triplets of bases, we give the name codons. Aha! In the language of messenger RNA, codons consist of 3 consecutive nucleotides, and we call them codons. Now, when you go about the business of transcribing something, it gets a little bit confusing because you're copying a strand of DNA called the template strand, which contains the DNA bases. You would make from that the complementary RNA bases, which don't have, remember, RNA doesn't have thymine, it has uracil. Well, that means that you can have uracil in the messenger RNA, but not the DNA. And then, get this, there's another whole type of RNA molecule that brings the amino acids to the ribosomes, and it has another thing called an anticodon. Now, I'm not going to ask you to memorize all of that because it really does get confusing. Instead, we're going to focus just on the messenger RNA triplets of bases, the codons. If we arrange this into a table, I'll give you a second to just look closely at this. Along the left edge of this diagram, we have the first base, which could be uracil, cytosine, adenine, or guanine. Remember, there's no thymine RNA, and the first letter stands for the name of the. The second base, if you look at the top of the diagram, also could consist of uracil, cytosine, adenine, and guanine. And along the right side of the diagram, it says third base, which again can consist of uracil, cytosine, adenine, guanine. And since this is the third base, we have to list it each time. UCAG, 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 all the way down. This is called a table of codons. If you look closely, each triplet of bases in messenger RNA codes for one of the 20 amino acids. For instance, if the three bases were uracil, uracil, and uracil, that would code for the amino acid phenylalanine. If the triplet of bases were cytosine, uracil, uracil, that would code for the amino acid leucine. If the triplet of bases were guanine, cytosine, uracil, that would code for the amino acid alanine. There are 64 different triplets of 3 nucleotide bases that you can make, but there's only 20 different amino acids. That means that in this table, there's a lot of duplication. If you take a look at leucine, well, there's 6 different combinations of bases that code for leucine. UUA, UUG, CUU, CUC, CUA, CUG. Any of those 6 would code for the amino acid leucine in the protein that is eventually to be assembled. It's also worth noting that there are 3 nucleotide codons, triplets, that say stop synthesizing RNA. That is, you don't need to put any more amino acids into the protein for this gene. Those triplets are UAA, UAG, and UGA. If you look at the table, it doesn't list an amino acid there, it just says stop. That means end of the protein. And again, there's duplication here because we have 64 triplets of bases and only 20 amino acids. In addition to the stop signal, there's also a start signal. Most proteins start with one particular amino acid, methionine. The signal for start is the triplet AUG. That's typically the amino acid that begins all proteins. Now, it may get cut in the final, proteins do get processed before they're ready-made and ready to do their job in the cell. So the methionine may get cut off, but that's the start signal. Okay, so of the 64 triplets, 61 of them correspond to an actual amino acid. The other 3 are stop signals, UAA, UAG, and UGA. Oh, and I should mention this. You'll never, ever be asked to memorize this table of codons, or I would never ask you to memorize the 3 stop signals. You'll always be given this table or something very similar to it on a test or a quiz. And that also includes not just tests in this class, but even the ACT or any other standardized test. They're going to give you this table. There's no reason to memorize it. Okay, let's try this problem together. We have a sequence of amino acids that are made from this DNA sequence. So here's a sequence of DNA, and it consists of this sequence of letters, TAC, and I'm going to divide these into triplets, okay? So I'm just drawing a little line here to say, okay, let's chunk this. TAC, CCT, CAA. Now remember, in transcription, DNA gets transcribed into messenger RNA. So what we're going to do is write the complementary base. So what's complementary to thymine? Adenine. So let's write an A for the messenger RNA base. Then we see in DNA, adenine. What's complementary to adenine? Be careful. If you were tempted to say thymine, you'd be correct if you were working in DNA. But remember, RNA has the base uracil, abbreviated with a U, in place of thymine. So uracil is complementary to adenine. And then C, well, cytosine is complementary to guanine. Okay, so we've got our first triplet transcribed. Let's draw a little line. Then our next triplet in DNA is CCT. Okay, in messenger RNA, we'll write the complementary bases. G, because guanine is complementary to cytosine, so CC means GG. Thymine, what's complementary to thymine? Adenine. Adenine exists in messenger RNA, so our next triplet is GGA in the language of messenger RNA. The third triplet, CAA. Well, guanine is complementary to cytosine. And then what's complementary to adenine? Be careful, there's no thymine in messenger RNA or in any kind of RNA. Instead, write a U for uracil. So our third triplet reads GUU in the messenger RNA. Okay, now let's identify the amino acids that would be produced from these codons. Here, we're going to look at our table up above. It's a codon table written in the language of messenger RNA. So that's important. And I'll never show you an anti-codon table, even though they exist. This is RNA, messenger RNA codons. All right, so AUG. If you find that one, you'll see here it's methionine, the start signal. MET is the abbreviation for methionine. Okay, next one, GGA. Look up in the table. Can you find GGA? Aha, here it is. GGA, glycine, whose abbreviation is GLY. Third one, GUU. Look up in the table. Can you find GUU? Aha. GUU codes for the amino acid valine, abbreviated VAL. So our sequence of amino acids from this original DNA sequence is methionine, glycine, valine. Now, students, most proteins are thousands of amino acids long. So maybe we put ... meaning this is going to continue for a long ways, but we don't want to be too tedious here. So we would just say and so on, or carry on. Okay, we did a lot with transcription. Now let's talk about translation. Once you've got messenger RNA made in the nucleus after transcribing the template DNA strand by the enzyme RNA polymerase, the messenger RNA needs to leave the nucleus in order to get to a ribosome. Ribosomes are the factories where proteins are actually made. There's another RNA molecule. mRNA is the messenger RNA. tRNA stands for transfer RNA. Its job is to bring the amino acids that are called for by the messenger RNA. It's a bit like the messenger RNA is the recipe for your favorite cookies, and the tRNA is the person who's dumping the ingredients together in the bowl and then mixing them. Well, guess what, students? There's 61 tRNA molecules, one for each of the amino acid codons. The tRNA bases, which are the same as the messenger RNA bases, only complementary, well, they form triplets that are called anticodons. Yes, there is an anticodon table, but I'm not going to have you use it because I think it's kind of confusing. Here's a nice full picture. Take a look at this. We've got 2 strands of DNA, a complementary strand, and the strand that's copied is called the template strand. Well, if the template strand has, actually, this is the same as our example. We did this together. It doesn't start until it gets a start signal. And that start signal, well, that was AUG, well, which is the complementary messenger RNA base to the DNA base is TAC. The second one was CCT, that's our next triplet. That transcribes to GGA in messenger RNA molecules. And our third triplet, CAA, would transcribe to GUU. And then we have to say etc. because there's going to be a whole lot more after this. Now, the messenger RNA interfaces with transfer RNA through another complementary pairing of bases. In messenger RNA language, if the triplet is AUG, well, the anticodon in tRNA is UAC. And if the next triplet is GGA, the anticodon would be CCU. And if the third triplet is GUU, the anticodon for that is CAA. Notice the similarity between the anticodons in transfer RNA and the original DNA sequence. Wherever there was a T, it's replaced with uracil for U, but other than that, it's exactly the same. It's the transfer RNA that brings the amino acids to the table, methionine, glycine, valine, and then continuing on. Okay, quick look at what actually happens at the factories, and then we'll be done with this note set. Okay, a ribosome consists of a portion that holds the messenger RNA in place. It does the job of reading the message. Then there's a place where the transfer RNA molecule with a linked amino acid binds and is held together in place. And there's another binding site so that another amino acid can come and be brought close to the first one. Take a look at this. If our first 2 amino acids were methionine and valine, well, we can see the codon in messenger RNA AUG and GUU and their corresponding anticodons here. Well, if methionine and valine are brought close to each other, there's a place where a peptide bond is likely to form. That's called the catalytic site. When methionine and valine are linked together, you've made the first link in a long chain of proteins. 2 transfer RNA molecules get brought close together. The amino acids are held together at a catalytic site to form a peptide bond, and then repeat many, many, many times. And let's say you've got a chain of a few hundred amino acids, or maybe even a few thousand. Well, eventually you're going to hit that stop signal, a stop codon such as UAG, which doesn't code for an amino acid. It just says you're done now. You can disassemble. The ribosome actually detaches from the messenger RNA molecule, and it goes off floating in the cytoplasm. Oh, one other common question is, where do the amino acids come from? Well, students, they're floating around your cells constantly. All 20 of the amino acids that are needed to make a protein are around in your cytoplasm, originally gotten from either your diet or other cells that manufacture them. They need to be around.